Good afternoon, everyone. Today, I'll be presenting our new method, dcHiC, an application for differential compartment analysis of HiC datasets. My name is Jeffrey Wang. I'm a high school student in the iLab at the La Jolla Institute for Immunology. Okay, so to begin, some background on this presentation. Even though this is pre-recorded, I assume at this point that HiC has already been thoroughly introduced and covered by previous presentations, that it is an assay to map the 3D chromatin conformation of the genome. And using HiC, we can derive heat maps that show us relative levels of interaction between different loci. When looking at observed versus expected levels of interaction, we see a plaid-like pattern, which reveals that the genome is spatially segregated into two distinct compartments. The A compartment, represented by red areas, which represent areas of enriched interaction, and the B compartment, represented by blue areas, which signify areas of depleted interaction. We can get a one-dimensional signal for compartments by performing principal components analysis on the observed versus expected correlation matrix, where the sign of the first eigenvector determines compartments. In this case, one sign correlates with the A compartment, whereas the other correlates with the B compartment. And although I won't get into this in a ton of depth, compartments have a wide range of biological importance. In particular, compartment changes in the actual 3D organization of the genome, are implicated in a wide variety of diseases, including cancer, including genetic disorders, and others listed here. As such, a better understanding of how compartments change can help us elucidate the mechanisms behind these diseases. In this presentation, I'll be going over our method to quantitatively compare compartments in order to find biologically significant differential interactions. To do this, I'll first go over some of our methods, solutions to existing roadblocks for compartment comparisons, before then giving an overview of our pipeline, and finally going over some results. So to begin, the primary motivation of our application is that eigenvectors by PCA are inherently not comparable. So to sort of conceptualize what I mean by this, let's say I have these two different cell lines or two different HiC datasets. For each one, I have these compartment signals. As you can see, there are two bins with matching values here, positive 2.5 and negative 3.1. However, even though these bins have compartment values that have the same sign and the same magnitude, they don't necessarily reflect the same degree of compartmentalization. The reason for this is that every HiC dataset harbors some biases, some of which you can see right here. And since PCA is just an unbiased transformation, those biases will remain in our final results. As such, the quantitative significance of eigenvector profile will change between HiC dataset, making direct comparisons between these values somewhat risky. We also see with PCA that the signs and the scaling can be somewhat arbitrary. So for example, for signs, we see that in this particular example that the signs can change between programming language and between cell line, posing an obvious roadblock to A/B interpretation. And the scaling also tends to vary as well. While this might not be a problem for pairwise comparisons or comparisons between small numbers of cell lines, it does become a problem when we want to take many different comparisons, 10, 15 cell lines. It can quickly become a very confusing factor. Our solution to this is a variant of PCA called multiple factor analysis, which normalizes for biases within multi-table datasets. So in multiple factor analysis, say we have k different HiC datasets, illustrated right here. The first step is to perform an individual PCA on each of these datasets. From that, we record the first eigenvalue, which can be thought of as a measure of a particular dataset's variance. We then normalize each data table by dividing it by its first eigenvalue, which will adjust the first principal component's length of each dataset to 1. And this essentially makes it so that no dataset can dominate any others. We then concatenate the adjusted data tables and perform a final PCA on the combined result. The result of this PCA we can then use to get the compartment signals for each individual HiC dataset. By using MFA and this normalization process, we obtain final PC values that have the same signs, the same scale, and comparable values. MFA also has a hierarchical extension called HMFA. In HMFA, we perform a succession of MFAs by going bottom up within some hierarchical tree. So for instance, let's say we have these 5 different cancer cell lines. 3 of them are breast cancer cell lines and 2 of them are lung cancer cell lines. And in HMFA, we first perform an MFA among the breast cancer cell lines, then one among the lung cancer cell lines, before finally performing an MFA between these two groups. In an HMFA, not only is every cell line normalized against the other cell lines in its respective group, the groups are also normalized against each other. And our application uses HMFA, meaning that this is extendable across any number of groups and an infinite number of cell lines. And this provides a lot of utility in situations where there is some intrinsic grouping within the data that we want to compare. Speaking of Hi-C comparisons, MFA or HMFA can be used in a wide variety of situations. So one such example is in time series data. So when we want to be able to track compartment changes throughout cellular differentiation. In this particular example, we're looking at embryonic stem cells to neural progenitor cells to cortical neurons. And we want to be able to see where the compartments change as the embryonic stem cell goes to the cortical neuron cell. Another example is just in large cohort-based comparisons. So one such example that we've performed that we mentioned in our abstract is a 57 cell line comparison between cancerous cells and normal cells. While we don't have time to go over the precise results from this comparison in this presentation, we will be going over results from this mycinal differentiation example later on. Okay, so at this point, we have balanced principal components using multiple factor analysis. Now the question is, how can we call biologically significant differential interactions within a variety of situations? So intuitively, the first thing you might want to do is to assign p-values to each bin. So our application does this through a measure called Mahalanobis distance, which is essentially a multivariate extension of z-score. And Mahalanobis distance squared is chi-square distributed, meaning that we can use this distance to find the distance between a point and the center of its distribution and convert it into a p-value. So in this particular example, we have the PC values of two cell lines graphed against each other. As you can see, most of the values hug this middle line. And this middle line, of course, representing areas where the PC values between both cell lines are equal. However, we see that we have a high Mahalanobis distance in points like these two yellow ones right here, where one cell line has a PC value of -1 and another cell line has a PC value of 1. These are the points that we want to be able to find because these ostensibly represent some significant biological difference. And using Mahalanobis distance, we're able to find all outliers within the data, which means that we can identify points like these. The only issue with just using M distance is that it just finds outliers, meaning that it will also capture fringe low or fringe high PC values that are not necessarily significant. In this case, these values are fairly similar across both cell lines, and these are not ones that we want to call. As such, we want to be able to distinguish between what we call technical variation, points like these, and biological variation, points like these yellow ones that represent real areas of interest. In order to do that, we quote unquote learn technical variation between pairs of replicates. So in order to do this, we take pairwise comparisons of replicates and graph their PC values against each other. We then fit linear models to each of these and calculate the distance between each of these points and the center fitted line. We then do this across all pairs of replicates and calculate the mean and the standard deviation of these replicate-wise distances for each chromosome. From here, we create a secondary measure we call DZ score. And this is used in comparisons between two different Hi-C datasets or two different cell lines. In this case, for a particular bin I, we take the point that that bin corresponds to, take the distance between it and the center fitted line. We then subtract the mean replicate-wise distance per chromosome and divide that by the standard deviation replicate-wise distance per chromosome in order to get the DZ score for that bin. In this case, DZ score will be higher for points that lie further away, and it'll be lower for points that lie closer to the center line. Now, as you can see, here we've combined both of the measures. With Mahalanobis distance, which is defined by the size of the bubble, we see that points that lie very far away from the line do have a large Mahalanobis distance. However, points that just have an extreme high or an extreme low value also have large distances. But with DZ score, we can get a better idea of which of these points are important and the ones that we want to call. We combine both of these measures in an independent hypothesis weighting, which is a variant of the Benjamini-Hochberg procedure that takes p-values derived from the Mahalanobis distance, but adds a covariate of DZ score, which will increase power in our comparisons. And as you can see in our final results, The size of the bubble here is represented by the log 10 of the p adjusted value. And as you can see, these points that lie outside of the bounds of replicate-wise technical variation are the ones that we call as biologically significant, ostensibly the ones that warrant further investigation. I should also mention that although all these comparisons have been done between pairs of cell lines here, these comparisons in our application are extended to general multivariate comparisons. So this is not necessarily just for 2 cell lines. Okay, so at this point, we have shown a method to normalize biases between Hi-C datasets and to get balanced principal components, as well as to call significant differential interactions between them. I'll now give a quick overview of our pipeline. The input to dcHiC are observed versus expected correlation matrices from a variety of sources, as well as a specified HMFA hierarchy and the chromosomes to be processed. We then perform HMFA, determine the sign of the principal component by aligning it against GC content or transcription start site locations, and then correct for cases where PC1 summarizes chromosome arms and PC2 should instead be used for compartmentalization. Our application then performs differential calling, as I specified earlier, and outputs summary and visualization files. Visualization is done through IGVR, which allows us to create these beautiful, interactive, standalone HTML files with gene tracks. As you can see here, we've done a multi-comparison, something more than just a pairwise comparison. And this track helps us easily identify areas of large compartment changes. We can do this for a variety of chromosomes. Performance-wise, our application is relatively computationally expensive, as it requires performing many different PCAs in a multiple factor analysis. However, if time is an issue, we do offer a multi-threaded processing option. Below, this is one example of time and memory usage in one particular instance that it was run. Okay, so finally, I'll talk about some results. These results were from the myostinal differentiation data I alluded to earlier, with an alpha level of 0.01 and a resolution of 100 kilobase pairs. The first thing we'll see is that dcHiC identifies many confirmed compartment changes. So in this particular instance, we have the DAPA4 gene, which is a gene associated with pluripotency in embryonic stem cells. As you can see, given the function of this gene, it makes sense that it is in the A compartment for MESC, but in the B compartment elsewhere. There is a large significant call to indicate the difference. And we also see this trend in LAMIN-B data, which measures how much this particular gene associates with lamina domains near the nucleus. In this case, there's a low level of association for embryonic stem cells, but a high level of association for neural progenitors, which indicates that this is within heterochromatin for neural progenitors, but it is more freely expressed within embryonic stem cells, confirming the compartments that we see up here. In addition, this is by far and away the largest compartment change in chromosome 16. We see a similar trend for the NANOG gene, which is a gene that encodes for a master transcription factor that is involved in pluripotency. Again, we see the A compartment versus the B, or just a rather low compartment. We also see a similar trend in the LAMIN-B data. And looking at our p-adjusted values, this is the 4th most significant call within chromosome 6. Finally, one last example. This one is for the KDM4C gene, another gene that has been shown to be necessary for differentiation of embryonic stem cells. It's in the A compartment here, but it's in the B compartment elsewhere. Again, a rather significant call, and the same trend within the LAMIN-B data. As you can see here, this is by far and away the largest call in chromosome 4. Okay, so in terms of genome-wide data, we can see that when analyzing regions of greater than 400 kilobase pairs, the change from embryonic stem cells to neural progenitors is the most pronounced, both in terms of the total number of significant regions called, as well as the number of base pairs they encompass. Another interesting thing that we see is that comparisons, significant differential interactions occur both in areas of compartment switches and in areas without compartment switches, which provides a future pathway, an exciting pathway for future analysis. Right now, compartment analysis is typically just done by looking at positive to negative. Is there a sign change? However, with our application, with comparable PC values, we can identify for example, a high A compartment to a low A compartment, which is a level of subtlety that currently does not really exist in compartment analysis. So one such example of this is right here, where we have a high A compartment in embryonic stem cells to a lower A compartment in neural progenitor cells and cortical neurons. And within this, we see that there's one particular gene called USO1, which is a gene that encodes for a Golgi vesicle transport factor. This is necessary for general cell function, which explains why it's in the A compartment everywhere. However, in literature, it has been reported that this particular gene is indispensable for embryonic development, which explains the high degree of compartmentalization within embryonic stem cells. Within RNA-seq data, we see a similar trend where it is more highly expressed in embryonic stem cells than the other two. Okay, so to conclude, I'd like to acknowledge my mentor, Abhijit Chakraborty, as well as my PI for Hot Eye, as well as all other iLab members in the La Jolla Institute for Immunology. If you'd like to try our tool, please head over to our GitHub and give it an install. And with that, thank you for listening, and I'll now take questions.